Yeah, thank you for the nice introduction. I hope you can all hear me well. Maybe I close the door here. So in the next 1.5 hours, my colleague Alexander Manet and we will present you or give a short insight into the microbiome research done here at the Medical University of Graz. As already mentioned, we are working at the Medical University of Graz in the group of Professor Christine Meusel-Eichinger. And our main projects or our main research focus lies, of course, on the microbiome and especially the archaeome. So not just the bacterial and fungal community, but also the archaeal community that lives on the one hand on and in our body. So we have samples from upper and lower respiratory tract, from oral cavity, from gastrointestinal tract, but also skin samples. And we want to decipher the role of these microbes that we find there in different conditions, such as, for example, human olfaction, inflammatory bowel disease, or cancer. But on the other hand, we also have some indoor environment projects like the hospital microbiome projects or the microbiome of plants and in space. For example, we had where we also worked on the project from the Mars mission and also got samples from the International Space Station. The samples that we usually get, or I will just focus in this presentation on the human part. So the samples for the human body parts that we usually get are, for example, stool samples, appendix samples, skin samples, nasal mucus, lung tissues, blood or saliva samples. And we have also people or participating in our studies from newborns to the elderlies. For the indoor environment, we sample surfaces, plants, the floor, different devices, for example, the PC or the mouse, and of course also humans that live in these specific environments. The general questions we want to answer with our microbiome research are: who is living there? So what microbes are there in this specific habitat? How many of them are there? What are they capable of, and what are they actually doing? And one of the bigger questions is always that we want to compare specific groups, for example, healthy and diseased participants, and want to see if there are differences between those groups. So why do we present microbiome research at the spring school? And why is, or should it be also important for you? Because we also have some projects together with the Institute of Psychology. For example, one of them, Fishy already mentioned, it's the sense of smell and the nasal microbiome. It's also called the nose-brain axis, where we want to see if there are differences between normosmics and dysosmics, because we want to see if the nasal microbiome, and when I'm speaking about nasal microbiome, I don't mean the whole microbial community that lives here in the nasal cavity, but the microbes that are stuck here in the mucus layer, directly next to the olfactory receptor cells. On this slide, I'm showing you some microscopy images from the scanning electron microscope. Now, as I'm showing you this image, I think no one of you will see bacteria. But here on this nasal epithelial cell, you can see a bacteria-like structure sitting right on it here. For this project, we sampled a lot or collected a lot of metadata. For example, we had the microbiome that was collected from the nasal cavity right next to the olfactory area. We also had stool samples, but we also had parameters like dietary nutrition MRI scans, blood samples, and we also had a longitudinal aspect by the smell training. The second project is the ocean rest study that also will be mentioned by Alexander Mahnert after my talk. So in the next talk, it's kind of the gut-brain axis. And our hypothesis here is that the personality mirrors the microbial composition in the gut, so that there is kind of a correlation or connection between our personality and the gut microbiome. The data we generated here was, again, from the stool microbiome. Then we had dietary information as well as psychological and neurological parameters like MRI scans and personality questionnaires. And now we want to combine these results from the different fields to show a bigger picture or get a bigger picture and correlate them with the personality with the microbiome. In the next few slides, I want to give kind of a plan what you have to think of before you want to start a microbiome study, because I think most of you never worked with microbes or did microbiome studies before. And the first thing is always, as in all other studies, you need an appropriate cohort. This means you need a specific sample size, you need controls. So for example, if you want to sample deceased persons, then you also need a cohort that shows for example, healthy controls. You need specific metadata information, of course, and the more, the better. The next thing is the sampling. You always have to decide beforehand, of course, which sampling area you want to choose. For the human body side, there are a lot of niches. Even within the nasal cavity, there are niches where the microbiome composition differs. So if you want to do an olfactory nasal microbiome study, then you should choose maybe the nasal microbes that are located directly next to the olfactory area. Of course, we have stool samples or also urine samples. The next thing is the sampling technique. So which devices you need for your to collect your samples. Here I show the normal swabs that all of you might know from the corona tests. Then we have, of course, the stool collection tubes and the nasal sinus secretion collectors. So we used, for example, for the nasal study, these sinus secretion collectors because we wanted to avoid any contamination that comes from the anterior nares. The next thing is the sample volume. You have to think about the sample volume before, because then you can decide which kit, for example, or which methods are appropriate for your specific samples. And of course, if they are high or low in biomass. For example, the stool microbiome is of course a high biomass sample, and the urine samples, for example, are low biomass. Then you have to pre-process your samples. And there are also many decisions that have to be made. So is it necessary to pre-process your samples? For example, with PMA treatment that just gives you the information if the cells are intact or not. So there are also methods where you need the pure samples or just a suspension of them. And if you have to put your samples into glycerol, for example, if you want to cultivate the microbes after the freezing and the storage again. Then the storage. The storage at the medical university, we are able to store the samples for long-term storage, for example, by minus 20 degrees or minus 80. We also have liquid nitrogen container where we can store them. And of course, they are also just for short time storage. You can just store them at a normal fridge at 4 degrees. Depending on the methods you are going to choose for downstream analysis, you always have to decide beforehand about the sampling technique, how you store the samples, and the pre-processing. And of course, pre-testing your methods is always very important and shouldn't be underestimated. The methods that we are usually using in our labs are listed here. So we are beginning with the samples. So we have human sampling material like stool or urine samples. Then we have also microbial cultures, enrichment cultures or co-cultures from our samples. And then most of us start with the DNA extraction and with downstream amplicon sequencing, qPCR or shotgun metagenomics. And with those techniques, we are answering the question, who is there, how many of them are there, and what are they capable of? To answer the question, what are they actually doing, we have two methods shown here, with the RNA-based metatranscriptomics and the NMR spectrometer for the metabolomics, to see what are they actually doing in their environment. Then we also have, of course, the cultivation, where we want to do microbial enrichments of our samples. And from here, you can either do some microscopy experiments, but you can also go again up here and start this workflow again. To just give you a small insight into the basics of our wet lab part of the microbiome research, I picked the standard procedure, so from the sampling material, DNA extraction to the basic amplicon sequencing. Most of our projects include this amplicon sequencing here, so to get the information on who is there, so the taxonomic information. First of all, of course, we have to analyze or isolate the DNA of our samples, and here we mainly use some kits, so there are really a lot of kits out there from different companies and for different purposes. For example, there are stool microbiome kits out there, or for saliva, for blood, and so on. And therefore, you have to choose wisely which of those kits should be used in your study. The next thing is to amplify a specific gene region. In our case, it's most of the time the variable region of the 16S rRNA gene to see the bacteria and the archaeal community, or the ITS region that is specific for the fungal microbial community. And then, of course, you have to sequence these fragments, for example, via the Illumina platform. And afterwards, the bioinformatics starts. So you come from the sampling to your raw data, and then, of course, to the visualization of your data. But this part will be, I think, discussed in more detail in the next presentation. So thank you for your attention, and I now...